Hello everyone and welcome to Felix's Space Time. In today's video I've got an interview with Eliah Overbey, an Assistant Professor of Bioastronautics at the University of Austin. She previously worked on the Inspiration 4 missions and are currently working on the Polaris Dawn mission and Axiom 2. If you like this video, don't forget to like and subscribe. Enjoy. First of all, thank you so much for agreeing to come on here for an interview today. I've got some questions and I'd love to get your kind of opinions on some things. For those who might not know you, could you tell everyone a bit about yourself?
Yeah, so my name's Eliah Overbey. I have a PhD in genomics from the University of Washington. Before that, I studied computer science. I became really interested in bioinformatics when I was doing computer science, and that's what led me to get my PhD in genomics. Then when I was in grad school, there was another student that put a pipette in my hand, and I learned how to do bench work and experiments, and built a lot of the skills that I use today.
Amazing. Before we kind of talk about your current research, the work you're doing now, you were an intern at the NASA Ames Research Center in 2019. What work did you carry out while you were there, and did it prepare you for the work that you do at the moment?
Yeah, so when I was at NASA Ames Research Center, I was there for a few months over the summer, and I was doing some work on retinal tissue from mice from the International Space Station. So there was a data set from mice from the RR9 mission that had bulk RNA sequencing done on the samples. We were curious what changes in gene expression there were in the retina. My collaborator, Vivian Mao at Loma Linda University, she had these amazing images of the retina from spaceflight from these mice, where it looked like there was cellular degeneration occurring in some of the immunofluorescent staining that she had done. And so what we saw with the bulk RNA-seq data was that we saw signs of retinitis pigmentosa, which is a cone rod photoreceptor degeneration disease. So we're able to pick up some of the signatures that we were seeing morphologically within the gene expression data.
All right, wow, some very advanced research there. So you are an Assistant Professor of Bioastronautics at the University of Austin. Can you tell me more about the work you do there? I also saw a piece posted by the university the other day about designing novel programs for students. I'd love to hear a bit more about that as well.
Yeah, absolutely. So yeah, I'm at the University of Austin in Texas. We're the newest university in America. We have our first undergraduate class starting this fall, 100 students. And so a lot of my work right now is centering around developing some foundational STEM curriculum for these students. But where we're going with this, and part of it was inspired by the Inspiration 4 mission, which I know you've talked extensively with all of those crew members, is that there's new pathways emerging for access to space. So Inspiration 4, you have a private individual who is able to fund their own mission independently of going through NASA. You know, in the past, every single science experiment, every single entrepreneurship project, they all went through NASA or through CASIS, which is the organizing institution for the ISS National Lab. So with the Inspiration 4 mission, it opened this pathway where people can access space through different avenues. So like Jared Isaacman went to SpaceX and commissioned the mission that way. And we're seeing this emerge also with like Axiom Space, who's now sending crews to the space station, but they're private crews. There's also an organization called SARA, the Space Exploration and Research Agency. They're commissioning Blue Origin missions and putting together crews to fill those missions. And so the idea is, can we get this set up so that it's now a university commissioning these missions, so that it's the students directly that are flying with their own research and their own ideas, and the faculty and the grad students? What could a program like that be?
Amazing. How did you first hear about the Inspiration 4 mission? I know it was their first kind of advertisement came out during the Super Bowl. Was it separate from that? How did you first hear about them?
So I first heard about it the first day of my postdoc when I joined Chris Mason's lab. I joined the Mason lab. I got a postdoctoral fellowship from NASA. I'd mentioned I did some bulk RNA sequencing work with and data analysis for retinal tissue in mice. So I wrote a postdoc proposal to do this at the single cell level, so you could tease apart gene expression differences and specifically different cell types in the retina. The retina is actually a very complex tissue with a lot of different cell types. And I wanted to follow up on that. But when I actually got to the lab my first day, Chris sat me down. We had a meeting with some of the leadership at SpaceX on the human research team, human operations team. We met with them. And they explained the mission to us, and we started to get to work on implementing the sample collection protocol that we did with the crew.
Amazing. I've kind of, as you said, I followed along quite closely with the Inspiration 4 mission from the beginning and knew the crew quite well through my interviews. So I'm very interested about your work with them in particular. I understand you published published research to do with their mission a few days ago. Could you talk a bit more about that, or why is that research so important?
Yeah. So throughout the mission with the crew, so we started collecting samples with them before their flight. We wanted longitudinal data pre-flight to collect a base flying from them. We collected as much as we could during flight. That was limited, though, because the Dragon capsule doesn't have all the resources that the ISS has. But we were able to get a subset of the samples that we collect on Earth in flight. And then immediately after they landed, we collected a full collection of samples. Immediately is within about 24 hours, as close as we could get. And then we did some post-flight longitudinal sampling as well, a month and 3 months out. And so what we were doing here, we were collecting blood, urine, stool, saliva, skin swabs. We got environmental swabs of the Dragon capsule during flight. We also got skin biopsies from the crew. So these were small pieces of skin from their deltoid region that we could do gene expression work on.
Amazing. And how does this from taking these samples, what happens to these samples after they've been taken? Can you talk me through that kind of process?
Yeah, so at each site that we do the sample collection at, we have to find a lab that we can partner with. This is because we have to spin down the blood, isolate the plasma from the red blood cells, from the PBMCs, the immune cells that we're processing. We need to do that fresh. We can't freeze the sample and do that back at Cornell. So we worked with the lab at USC when we were in Hawthorne, visiting SpaceX on site there. We worked with another lab at Baylor College of Medicine during Houston sampling collections, and another lab when we were in Cape Canaveral. And so once we do all the processing work there to get the samples stable. Once they're stable, we send them back to Cornell so that we can standardize the processing there from that point on.
Amazing. So you're doing research with the Polaris Dawn mission as well. How did you first hear about them? Was it did you follow Jared from, he'd finished Inspiration 4 and he'd announced Polaris Dawn?
Exactly. Yeah. Since we were working on Inspiration 4, we heard about Polaris Dawn pretty early, from just, proximity and working on that mission.
Amazing. I'm quite interested in the Polaris Dawn mission as well. So they're launching at the end of this month, and you'll be there conducting research, I understand. What do you aim to achieve with your work with Polaris Dawn? Will you be doing the same kind of stuff you did with Inspiration 4, or are you adding some different bits to it?
So we're doing the same that we did with Inspiration 4. So what we're looking for in the Polaris Dawn crew is really increasing the sample size from the amount of astronauts that we've collected from. So all of this work started, or at least the first omics study, an astronaut was the NASA twin study, which was 1 astronaut in space for a year. Inspiration 4 increased our sample size to 4 astronauts. If you're familiar with scientific research, these are still very small numbers. It's really hard to generalize, what'll happen to an entire population if they go to space based on 4 people. So Polaris Dawn will allow us to expand that to, it'll be 7 astronauts total, the 4 from Inspiration 4, and then the 3 new ones from, that are flying on Polaris Dawn, and then we'll have Jared twice. So we'll pay special attention to what happens to repeat flyers, especially since they're going out further this time.
Amazing. You talked about, these were relatively small numbers. Are you looking into doing these samples on more space flights? I know you talked a bit about Axiom 2.
Yeah, so we have collected samples from the Axiom 2 crew. It was a slightly more limited profile, just because the dynamics of planning missions with Axiom is different than SpaceX, is different than NASA. So we have blood samples from them, we have urine samples from them. We do have a series of papers in review for those astronauts as well that we're currently trying to push through the peer review process.
Amazing. So this is a question from one of my subscribers. Given that all human spaceflight since 1972 has been in low Earth orbit, what new research into human health is required for the higher radiation environment of high orbit missions such as Polaris Dawn and future crewed missions to the Moon and Mars?
Yeah, that's a huge concern. Is the further out you go, the more radiation you're going to be exposed to. ISS astronauts are still quite protected compared to what people will experience on the moon or on Mars. From my perspective, I'm most worried about, from radiation, most worried about the cancer risk. So I'm interested in seeing, one thing we did with the Inspiration 4 crew was we studied what's called clonal hematopoiesis. So this is when we take immune cells. So we take the immune cells from their blood and we look for different variants there before and after flight. And we don't just look immediately after flight, we look months after flight as well. We're actually still collecting samples from the twin study and from the Inspiration 4 crew whenever they're in town, to keep following up on this. And what we do is we look to see whether or not there are certain variants overtaking the population of immune cells, because this would be indicative that there's some population that's growing out of control. So really, when we're looking at these extended durations and radiation, finding ways to monitor cancer risk over time will be paramount.
Amazing. So kind of with your research with Inspiration 4, you said you took samples beforehand, during the mission, and afterwards. What kind of did you see any changes in the samples you took? Can you talk a bit more about kind of the outcome of that research?
Yes. So one of the samples that ended up being really influential that we collected during flight was we got capillary blood from the fingertips. So we asked them to do, if you've ever been to the doctor and gotten like a lancet finger stick, we collect just a few droplets of blood from that. And we got them at a couple time points in flight, and then of course before and after flight. And what we can look at in those samples are telomere lengths. So telomeres are the like shoelace caps on the end of your chromosomes. What happens is as your cells divide, so as you age, the telomeres actually get shorter and shorter with each cell division. One thing that was noticed during the twin study and was followed up on some additional astronaut cohorts on the space station was that telomeres in certain subpopulations of cells in your immune system were actually getting longer. And so there was some question, to me was, I know high intensity interval training on Earth can have some impact on your telomere length. And astronauts are exercising 1 to 2 hours a day while they're on the International Space Station. So I didn't know if perhaps this was a side effect of the exercise regimens that they were undergoing to maintain bone and muscle integrity. What we saw with Inspiration 4 was, that capsule, that Dragon capsule, that's the size of a large camping tent. You're not getting your workout in during those 3 days. So what we saw with those samples was we saw the telomere length trending upwards in all 4 crew members, and then in one crew member, it was statistically significant during that period, but it returned back down to normal as soon as they returned back to Earth.
Amazing. And kind of that, you talked about the changing the length and it came back to normal. What does that show you about kind of what happened kind of during the flight?
Yeah, well, the interesting question, like follow-up question to me, is what was causing that lengthening? Because there's like conflicting, media narratives. If you've seen like press stories, they kind of like to push the story really far in either direction. So you'll see a news article that'll say like spaceflight is the fountain of youth because your telomeres are getting longer. But there's also forms of cancer that protect telomere lengths and cause them to get longer. So understanding what these mechanisms are and what they might do over time is going to be something to continue to monitor in these crews.
Okay. So my final question for you today, what are your plans for the future, kind of beyond Polaris Dawn and Axiom 2? Do you have any other missions you'd like to oversee? What's coming next for you?
Yeah, so the protocols that we have are open for any astronauts that are going into space. So I, hope that the future Polaris missions will continue to participate in the protocol and contribute to that body of research, any other commercial missions as well. It would be interesting to get some data from like Blue Origin and Virgin Galactic missions, even though they have a very short microgravity exposure, just getting the human body's reaction to takeoff and landing would be a good control data set to have to compare to these crew members when we're writing up future publications and doing these analyses. Beyond that, we're also, one of the things I'm working on building here at UATX is an astronaut genome sequencing center. So one of the issues that we anticipate having as this field of research continues to expand is where is the money going to come to continue sequencing astronauts? Because there's going to be an issue of scale, especially as we think about Starship taking off and have hundreds of people who could participate. So we're opening up a sequencing center here where the profits that we get from the sequencing core are going to be applied specifically to astronaut samples.
Amazing. Well, thank you so much for coming on here for an interview today. You had amazing answers to my questions. It was amazing getting to learn more about your research with Inspiration 4. And I can't wait to see kind of what comes with you for Polaris Dawn. For people who'd like to kind of to read more about your work, where can they find your stuff?
Okay, yeah. So we have, if you just type in SOMA, so our project's called SOMA, the Space Omics and Medical Atlas. SOMA and then space, I think you have to type space because other stuff comes up, Space Omics. You'll see our Nature portfolio package. So we had a collection of, there were 44 papers that came out pretty much the same day at Nature. At least 16 of them, probably more, covered specific facets of the Inspiration 4 mission. Some of it was NASA data, JAXA data, these other organizations. So that's a really good sort of point to dive into. Yeah, to get a primer to everything that's going on.
Amazing. Well, once again, thank you so much for coming on here for an interview. And I hope you have a wonderful rest of your day.
Thank you. Thanks for having me on.
Thanks for watching. If you enjoyed this video, don't forget to like and subscribe. See you next time.